So begin? All right. Hello everyone, I am Catherine Ryan, or Katie Ryan. I'm currently in the Summer Research Institute and doing research in the laboratory of Dr. Vishwanathan in the Department of Veterinary Science and Microbiology. Okay, so just an overview. The goal that my research is trying to achieve is to show that ESPF causes an increase in the degradation of EGFR. Last time I was saying how ESPF affects apoptosis of a cell, but we kind of went a little bit more detailed than that, how it affects the degradation of EGFR, which overall affects the apoptosis of a cell. Just to remind you, ESPF is a secreted protein from enteropathogenic Escherichia coli, EPEC. It's secreted through a type 3 secretion system directly into the host epithelial cell. And EGFR is the epidermal growth factor receptor. It's a protein that is bound on the membrane of your host epithelial cells. And in its inactive form, it's a monomer. Once it binds to its ligand, whatever that will be on the outside of the cell, it dimerizes, either hetero or homodimerizes. So that's just to let you know what I'm talking about. And again, I'm just going to go over the schematic that I did last time. So basically, this is what we believe is happening. We originally did research on a protein that's secreted initially in the EPEC infection, and that's ESPZ. That's basically, it's inhibiting all this from happening to prevent the cell from dying so that it's able to colonize your gut. Later on during the infection, EPEC will secrete ESPF, which actually activates this protein cytochrome C, which is bound to the membrane, which activates caspases, which we believe is degrading EGFR. This is important because normal cell processes in a healthy normal cell, when your EGFR is broken down, it's usually due to phosphorylation or ubiquitization. Basically, EGFR will bind to a ligand, and it's either recycled, it's phosphorylated and recycled back to the cell membrane, or it's tagged with an ubiquitin and sent to a lysosome and broken down. What we believe is happening is that ESPF's activating a cascade of reactions which activates a caspase, which is a protease. And the protease is breaking down EGFR rather than the normal cell processes. Okay, so here's a table of the different bacterial strains that I'm looking at when I infect my cells. First of all, we have our wild type. Then we have a deleted ESPF gene, and that's basically just a genetically modified wild type that we've taken the gene out, so it's unable to produce that protein. Then we have recomplemented that gene back into the cell. However, it's not able to be activated unless we add IPTG, which is basically just a lactose metabolite that turns on that gene to produce the protein. The more IPTG we add, so as you see we have 10 micromolars, 100, and 1,000 micromolars, the more protein it will produce. We also looked at a gene that deleted the ESPZ protein, which is the one that prevents initially that prevents apoptosis. And then we also did a complement ESPZ, which we just put that gene right back in somewhere else in the chromosome. So basically, first what I did was my tissue culture cell line was CACO2 BBE cells, which are human epithelial cells. I grew them in nutrient medium, DMEM, FBS, which is the serum and glucose heaps, just to maintain. These cells are very finicky, so they can phosphorylate EGFR with different osmotic pressure and different pH. So basically, we just made the medium perfect so that it would not do that. We grew them at 37 degrees Celsius in 5% carbon dioxide. And we grew them 7 to 10 days before we waited until they were 95, 100% confluency, which means that they're completely next to each other before we actually infected them with the bacteria. I grew them in 6-well tissue culture plates. So the day before the infection, I would change out the medium or nutrient-rich medium to serum-free medium, and I would grow my bacteria in LB agar overnight. In the morning, I then put my bacteria in serum-free medium also and grow them to 0.4 OD, which is about mid-log. That's when they're growing the fastest. I then would put the bacteria in the 6-well plates with my cell cultures and an MOI, multiplicity of infection. So basically MOI of 100 and MOI of 500 is 100 bacteria cells for every 1 human cell, 500 bacteria cells for every 1 human cell. And then I had them in a 37 °C for 1 hour. Then I change out the medium just to make sure that all the bacteria that hasn't attached to the cells is removed, and we're only focusing on the bacteria that's attached to the cells. And I put fresh media in and continue to allow them to incubate for another 4 hours. After the 4-hour time period, I then remove the media and I lyse the cells. I make sure that there's like a protease and a phosphatase inhibitor in my cell lysis, because we don't want any, like I said, the cells are finicky, so we don't want any of that happening after. We just want to focus on what the bacteria is doing to the cells. Okay, so after I lyse the cells, like I said, EGFR is a membrane-bound protein, so I have to sonicate them, and it's really tedious and it takes a really long time. So I do that for each 300 microliters. I sonicate them 30 seconds for 3 times each. And then once they're nice and sonicated, I go ahead and do a protein quantitation with the Bradford assay. And that's just basically making sure that we have the same amount of protein that I'm going to load in the gel, so that there's not like, look, there's more EGFR here, but I loaded twice as much protein. I just want to make sure it's the same across the board. And then I ran a SDS page, and basically that is a gel. SDS is a compound that will put a negative charge on the protein and linearize it, and it runs through the gel so that we could separate it by size. After I do that, I then transfer the wet gel to a membrane, to a nitrocellulose membrane via wet transfer. I do that at 100 volts for about 2 to 4 hours. And then once we have it on a membrane, I can actually probe it with an antibody that's specific to EGFR. So I will incubate it in my primary antibody at 4 degrees Celsius overnight. Then I wash it and incubate it with the secondary antibody and develop my blot. I don't have a picture of my blot, but I promise I do have it. This is what it should look like. So what you're seeing here is the mock infection is just the cells without any bacteria. We just put them in media just to show that nothing else is causing degradation of EGFR other than the bacteria. So you should just see the normal level of EGFR. In the wild type infection, you should see a decreased level. And what you see here is actin. Actin's just a normal protein that is in every cell. And we're doing an actin probe too, just to show that there's the same protein across the board with different levels of ESPF and EGFR. In our mutant, there's going to be no ESPF. So you're going to see the same level in the mutant as you do in the mock. Again, we're going to test how much ESPF is in the cell, and you should see none in the mutant. So here we have our complemented bacteria, where there's, actually this should say 10, 100, and 1,000 micromolars. And so you see as the increase of IPTG, the decrease of ESPF. Again, as the increase of you see the increase of ESPF. So this is what we should see, and I actually do have some blots that do show this information, which is super exciting, because not only it's going to get published for reals too, so that's awesome. So if you guys come see me at my presentation, you'll see the data too, and we have it. It's just it was broken up into different blots, and now we're just trying to make it look pretty so we can actually have it published. So, and I would like to acknowledge all the awesome people in SRI and UROC, and of course my mentors in the lab. And if you have any questions, here's my email, or you can ask me now.